can just get us started with some introductory remarks. Hi, everyone. I'm from Science in the News. Welcome to one of our lectures. This is our third lecture of the semester that we've been doing virtually, and they've been going really well. Thank you for joining us. So Science in the News is an organization based at Harvard. The goal is to get graduate students to connect to the general public with regards to their research. So it's both to get them to improve how to share their science, so improving science communication, but also get to share groundbreaking research with all of you. So the lecture series is only one of the things we do. I'll post a link to our website later in the chat if you want to see the other kinds of things. We have a blog that we constantly update with either new things, new scientific discoveries in sort of a general language that everybody can understand. Just logistics for today. So you all do not have the freedom to unmute yourself, and we will allow you to if you have questions. We have 3 sections. Each of them will take 15 to 20 minutes. And throughout the sections, we won't interrupt Ali, our speaker of the day. And then at the end, we'll ask her the questions. But as she's presenting, if you have questions that come up, feel free to write them down in the chat, and we'll relay them to Ali at the end of each section. So at the end of each of the 3 sections, we'll have time to answer questions, clarify things if you have any things that you're confused about. And there's also, so you might be listening through Zoom or through YouTube. We look at both of them. So we will answer questions both from Zoom and from YouTube. Obviously from YouTube, you can only type and we will ask your questions. I will probably read it out loud. Lily might read it out loud. In other cases, if it's on Zoom, you can ask to be unmuted to ask your question in person, or if you want a follow-up. Those are the logistics. Yeah, and I can also post a link to the YouTube as well. Sometimes Zoom can have issues. If you have issues with Zoom, YouTube might be an easy place to go if you have some technical difficulties and want to keep watching the lecture. And if you have any kind of technical issues, feel free to message me, Lily. We all have Science in the News in the beginning of our names, SITN. So feel free to reach out to us if you have any technical difficulties or specific questions with regards to the talk. Today's talk is super timely. We're incredibly excited. Ali is a wonderful Science in the News member.
This meeting is being recorded.
And also, this meeting is being recorded, as you've heard, so so that we can have it live on YouTube. You can go back and watch it later, too. Yeah, and Ali also very recently, 2 weeks ago, or is it only a week, defended her thesis, and she's now a doctor. She has her PhD, and she will share with us about COVID-19 and the vaccines, but I'll let her introduce herself a little further. So yeah, hope you enjoyed.
Thanks, Booz. Hey everyone, my name's Ali, and I, like she said, recently earned my doctorate in microbiology and immunology at Harvard, and I've been involved with Science in the News as a volunteer all through grad school, so I'm really excited to be here today to talk with you a little bit about COVID-19 and vaccine research as my last volunteer event with this really amazing organization. So, quick disclaimer before we get started, this presentation should not replace medical advice from a qualified physician. And if you're experiencing any symptoms of COVID-19, you should contact your local health authorities for guidance. If you live in Massachusetts, you can find information on testing and reporting on the mass.gov/coronavirus website. And the CDC website listed here has a very similar page with information applicable to everyone in the US. So this presentation will be divided into 3 parts. First, I'll give an introduction to the coronavirus pandemic. Next, I'll describe how the human immune system responds to viral infection and how we can use that information to design vaccines. Lastly, I'll talk about the current state of vaccines for COVID-19. First, onto an introduction. So let's walk through the sequence of events that brought us to where we are today in the pandemic. So in early December of 2019, doctors in Wuhan City, China, noticed an uptick in patients with a pneumonia-like respiratory illness. At the time, they didn't know what the cause of the disease was. The number of infections continued to rise, so on December 31st, the World Health Organization, or WHO, was informed of this outbreak, and an investigation began to find the cause. On January 7th, Chinese scientists isolated a new virus from a patient suffering with this disease and named it SARS-CoV-2. On January 11th, China reported its first death from this new infection. And by January 13th, the virus had actually left China, first surfacing in Thailand and then quickly in several other countries, Japan, Korea, and making its way to the US on January 21st. By the end of January, the WHO had declared a public health emergency. And by the middle of March, almost every country in the world had been infected with this virus. So I'm going to show you a short animation which I think really nicely illustrates the spread of this virus. So as I said, it was first identified in Wuhan City, China. And here we will see the total number of cases as it starts small and then explodes as the virus spreads around the world. Here on the bottom right corner, you can see the date in which these events were happening. And the circles represent the number of cases in that country, and bigger circles represent more cases. So we can see, we can see here that the virus started in China and then in early January spread first to Thailand and Japan and then into the US. Later in January, it spread across Asia, hitting Australia and Europe. And you can see here by early February, we're already getting cases in almost every country in Europe and Asia. And again, this number is just increasing really dramatically. You can see in early February, there's a huge amount of cases in China. And as February ticks on, we start to see the first cases in Africa, South America, and spreading through Europe. And as we let this animation roll, we will see that by the time we get to the middle of March, basically every single country in the world has become infected with this virus. And today, on December 2nd, 2020, we have had to date over 64 million total cases of SARS-CoV-2 infections, which have resulted in 1.5 million deaths. In the USA alone, we've had almost 14 million cases and nearly 280,000 deaths. And these numbers are really, truly staggering, and they're actually continuing to rise basically every day. And so, how did we get here? I want to take a step back in the next few slides and actually examine the virus itself and its properties, which allowed for such rapid and dramatic spread around the world. So SARS-CoV-2 is a virus responsible for this pandemic. Viruses are microscopic particles. They're so tiny they can only be seen with a powerful microscope. Here's a cartoon structure of the SARS-CoV-2 virus. And one of these virus particles is approximately 0.1 microns in diameter, which is about 100 times smaller than the width of a single human hair. And this is an image of the real SARS-CoV-2 virus taken with one of those powerful microscopes. And you can appreciate that these viruses have these little bumps along their surface. And as we'll talk more extensively later, these bumps or protrusions are the virus's spike protein. This protein actually allows the virus to stick to the lung cells and cause infection. And we'll also see later that this spike protein is a tool that we can use to educate the immune system on how to fight off this virus in a vaccine. So viruses like SARS-CoV-2 are considered to be pathogenic, which means that they can cause infection and disease in humans. It's possible for infections to spread from person to person. So here we have someone who is infected with SARS-CoV-2 and a person who's not infected, who's healthy. The infected person can transfer some of the virus through the air to the uninfected person and infect them. This person now has the SARS-CoV-2 virus inside their lungs and can go on to spread the infection to someone else. And importantly, viruses cannot survive forever outside of a human host. They're kind of like parasites. They require being inside of a person to multiply. SARS-CoV-2 is part of a family of similar viruses called coronaviruses. These viruses infect the lungs and cause respiratory illness. And coronaviruses include some other pathogenic viruses you may have heard of before, in particular SARS and MERS, which caused dangerous outbreaks in the past, as well as the common cold. And I want to make clear a couple of terms which you've probably heard about when reading about this pandemic. So to clarify, SARS-CoV-2 is the virus, a type of coronavirus to be specific. And infection by SARS-CoV-2 in the lungs causes COVID-19 or coronavirus disease. And so we can think about what happens when an individual is infected with SARS-CoV-2 and how the virus actually spreads from person to person. So this infected individual, through breathing, coughing, and sneezing, actually expels these watery drops from their respiratory tract into the air. These droplets contain the virus particles inside of them. And virus-containing droplets come in two forms. The first are these large droplets, which are actually relatively heavy and fall to the ground because of gravity within 6 feet of the person breathing. And scientists think that most transmission or spread of SARS-CoV-2 comes from inhaling these large droplets. There are also small droplets that can stay airborne for longer and travel farther than 6 feet. These droplets can also infect individuals through inhalation. Droplets can also fall onto surfaces like phones or household objects and contaminate them with the virus, but this actually seems to be a pretty uncommon way to acquire the infection. The most common, again, is to acquire the infection from close contact with an infected person within 6 feet, meaning that most infected individuals were infected by inhaling these large droplets within a 6-foot radius. And one reason why the virus has been able to spread so rapidly is that it can take a while for an infected person to realize they're sick. So let's walk through an example of this average person who starts out healthy, uninfected, who comes in contact with SARS-CoV-2. For the next 5 or 6 days, this person is infected, but it's unlikely that they will have any symptoms or feel sick. During this time, they have a risk of receiving a false negative test because there's just not enough virus in their body to actually be detected by the test. But unfortunately, during this time period, this person can shed virus into the environment and infect others unknowingly. Most people will start to feel symptoms at around day 5 or 6, but for others, it can take up to 14 days. And as you're progressing through this infection, after day 5 or 6, people will start to feel symptoms, start to feel sick, maybe develop a cough or a fever. And during this phase, there's enough virus in the body for them to be able to test positive. And of course, during this time period, they can also infect others. And the severity of the respiratory infection depends a lot on many factors, but in particular, age and health status seem to be contributors towards having a more severe disease. And infected individuals will remain infectious for the duration of their illness, which in mild cases is a week or 2, but up to 6 to 8 weeks for more severely ill patients. And so this incubation period or phase before the person knows that they're infected, before they start feeling symptoms, is very dangerous. People can be spreading the virus during this period. And so this is why it's really important to quarantine after a possible exposure as to prevent infecting those before you know you're sick. And this is also why it's good to wait a couple of days before getting a test after exposure to basically ensure that there's enough virus in your system to detect. And so you can imagine someone who's infected and shedding virus into their environment, coming in close contact with others, they could infect people. Now, these two who are infected can infect others they come in close contact with, and so on. And because SARS-CoV-2 is a brand new virus to the human population, there was no pre-existing immunity, meaning everyone in the world was vulnerable. And it is easier than ever in our hyper-connected world for a respiratory virus to spread extremely quickly. So this is an a map of all airplane routes traveled in 2019. And the darker red the color indicates more flights along that path. And so it's pretty obvious to see that our world is extremely connected. And early cases of SARS-CoV-2 infection in other Asian countries and the United States can actually be traced back to travelers coming from Wuhan City, China. And so I like to think that we have to take extra precautions when traveling now in order to stop the virus from spreading even further into communities that may not have been affected as severely. And so, what can we do to stop the spread of the virus? So far, the only tools we've had are preventative measures, which have actually been very effective at reducing risk in populations when used effectively. So, masks worn over the mouth and nose are one of the most effective ways to reduce the spread of the virus. Also, social distancing, which means keeping a distance of over 6 feet between individuals, can limit the ability of those heavy droplets to reach a new individual. Another preventative strategy is a vaccine. Vaccines are like shields. They block the virus from causing disease. And in combination with masks and social distancing, a vaccine will help bring this pandemic to the end. And in the next section, we'll talk in more detail how vaccines actually work and the immune response to infection. But I want to pause here to summarize before we move on. So there's been a new virus called SARS-CoV-2, which causes lung infection and the respiratory disease COVID-19. The virus has spread around the world rapidly, and cases are continuing to rise. Preventative measures like masks and social distancing have helped curb the spread of the virus. And I can pause here to take a few questions, and you can type them into the chat or comment on the YouTube, and we can chat for a little bit before, make sure we're on the same page before we move on.
We'll take a few minutes. There's also a short delay for the YouTube, so we usually try to wait. So write them in the chat. You can also ask in the chat to be unmuted if you have a particular question, then we can unmute you. Okay, we have one in the chat. I'll read it. It's from Tracy Moore. You mentioned that some can be infectious for 6 to 8 weeks. Is it possible to be infected and thus a spreader that long if you don't have any symptoms?
That's a great question. So I actually have a little slide here. So the CDC says that individuals who do not have symptoms are no longer infectious 10 days after their last positive viral test, assuming that they start to get negative tests. So people who are experiencing symptoms, that means the virus is, of course, still inside you causing disease. But for those who do not have who do not have symptoms, there's a little bit of a faster turnaround time. So I don't think it's possible for asymptomatic people to be shedding virus for like months. It's probably more on the order of like a week and a half or 2 weeks.
Thanks, Allie. If you have follow-ups, Tracy, feel free to put it in the chat. We have another question that asks, how long can the virus stay in the air?
So those virus-containing droplets are settling to the ground like relatively quickly, like within seconds, if not minutes. The smaller droplets can remain in the air for longer. I don't have an exact time frame because it's a little bit harder to study those. On surfaces, though, people have reported days of the virus being able to survive once like an object is contaminated. But it seems like most of the transmission is happening within close contact, meaning people are inhaling those large droplets and not from like touching contaminated surfaces.
Thank you for that answer. Do we have any more lingering questions before we keep learning? Oh, we have one more. And then we do a short break, so we'll do like a 5-minute water break as well. How stable is the virus to sunlight? This is an interesting question.
It's an interesting question, yeah. So UV light can destroy the virus particles. So I imagine that could come from the sun and over time destroy the virus, but it's a little bit hard to say exactly sunlight versus direct UV exposure. And I think probably the best answer is that to feel safe as possible, people have just been wiping down surfaces with antiviral wipes. But again, contamination from surfaces leading to infection is actually pretty rare. It's more transmission from close contact that we should be worried about. Yeah. Cool, maybe we should move on to the next part. And if you guys think of questions, feel free to write them in the chat and I can answer them at the little question breaks. So next we will be discussing how the immune response actually recognizes this virus and how we can sort of educate the immune system to respond better and more efficiently with a vaccine. So I want to walk through the steps of what happens at a cellular level during the infection with SARS-CoV-2. And I want to emphasize strongly here, the reason that we care about this is that understanding what happens between the virus and the human body during infection at a cellular level is what allows us to design effective vaccines. And I'll talk about this a little more later, but scientists have actually been doing research on coronaviruses for many years before the pandemic. And information that they discovered about other similar coronaviruses like SARS, and MERS have helped us have a more clear picture of what's happening inside the body. And a lot of the information I'm about to tell you comes from those types of studies. And so let's take a look inside the body during infection. Here we have a cartoon of a person who's not infected, and then they come in close contact with an infected person who is expelling these virus-laden droplets. The uninfected person breathes in some of these droplets and delivers the virus to their lungs. Inside the lungs, there are these structures called alveoli, which are little air sacs. And in a healthy lung, these structures are responsible for performing gas exchange. And this is a critical function of the lung. We breathe in oxygen and breathe out carbon dioxide, and those gases enter and leave the blood by passage through the alveoli. During a SARS-CoV-2 infection, the virus actually enters these air sacs and infects the cells lining the air sacs. This damages the structures. And as the infection progresses, damaged alveoli can actually fill with fluid, which leads to respiratory failure. It's impossible for the lungs to adequately perform that gas exchange when these structures are literally suffocating. And so let's zoom in now on one of these air sacs and see what's actually happening during an infection. And so here's how the infection really looks. This image was taken with a powerful microscope. These little yellow blobs are the SARS-CoV-2 virus particles. And this pink folded material is part of a human cell that's being infected. And here's another image so you can really appreciate the size difference between the virus and our human cells. This giant purple structure here is a single human lung cell isolated from an infected patient. And again, these yellow spheres are the SARS-CoV-2 particles. You can see just how much smaller this virus really is, yet it's completely overwhelming this cell and actually killing it. And so we can think of the interaction between the virus and our body as a microscopic or cellular war. SARS-CoV-2 virus is the invader, and our human cells are the defender. Our body is comprised of trillions of cells, and there are many different kinds of cells with unique jobs. And as I'll explain in the next few slides, there are several different types of cells involved in this microscopic war with SARS-CoV-2. So let's first walk through the different players that are involved in this cellular war and the tools or cellular proteins that they will use to do their job. So first we have the invader, the virus SARS-CoV-2. The protein tool important for viral success is this spike protein, which is covering the viral surface. And spike is very sticky, and it allows the virus to attach to the lung cells. Lung cells themselves play an important role as the first line of defense in the lung. They join together very tightly to form a barrier. But their main tool in this war is their ability to call for backup or display a type of SOS signal during infection, which we'll see is critical for recruiting immune cells to the site of infection. The cells that respond to these calls for help by the lung cells are the first responder immune cells, which are these purple kind of cool looking spiky cells. And their job is to recruit the second line of defense, which are specialized immune cells. These specialized immune cells break out the big guns to fight the virus, and these tools include killing molecules and antibodies, which I'll discuss in more detail in a few slides. So now that we know all the players, let's walk through what actually happens in the lung during infection. After droplets containing the virus are inhaled, the SARS-CoV-2 enters the lungs. It sticks to the lung using that sticky spike protein. Next, the virus actually enters or goes inside the lung cell. Here, it hijacks the cell's resources and machinery to generate components of the virus, which are all these little parts you see here. These parts can assemble inside the cell into new viruses. And these new viruses can then leave and go on to infect another cell in the lung and restart this cycle. So this activity of making virus is not a normal function of lung cells. As I mentioned before, the purpose of these lung cells is to perform gas exchange. So this cell on the right, which is not infected, is performing its normal function of delivering oxygen into the blood and moving carbon dioxide out of the blood. But infected cells, the lungs are preoccupied with making virus instead of doing this normal job of gas exchange. And they do this because when the virus infects the cell, it actually delivers a set of instructions, genetic material, for how to make more viruses. The lung cell follows these instructions and is essentially converted into little virus factories. And this preoccupation with making virus instead of performing gas exchange contributes to decreased lung function. So what can the lung cells do to get help during the infection? They have a few strategies. The first is to send out an SOS signal, which signifies to the immune system that they're infected and need help. Infected lung cells also move pieces of the virus that they're producing to the cell surface as a way to advertise to the immune system what type of invader is inside them. First responder immune cells in the lung see these help me signal and stop and pick up a piece of the invader, as you can see here. This cell carrying this piece of the virus then leaves the lung into the blood vessels to alert the second line of defense. The first responder cell travels through the blood and then arrives at the home of specialized immune cells, the lymph node. These cells are called B and T cells, and they have specialized jobs in fighting infection. When the first responder cell arrives, it shows the specialized immune cells what the invader looks like, sort of like a wanted poster, and encourages these cells to get ready to fight. And so the first thing the B and T cells do is multiply in the lymph node to create an army big enough to fight the infection. So B and T cells produce specialized protein tools to fight the infection. T cells, which are these green cells, make killing molecules, which I've represented here by these sort of Pac-Man-like circles. These molecules remain inside the T cell until they're ready to be unleashed onto an infected cell to shut down the virus factory. B cells make antibodies, which, as we will see on the next slide, function to actually block the virus from entering an uninfected lung cell. So T cells with their killing molecules leave the lymph node back into the blood to travel back to the lung. B cells stay behind in the lymph node but release their antibodies into the blood to go to the lung. So back at the lung, the T cells and antibodies work together to block viral spread. First, T cells arrive from the blood to the site of infection. They can identify the infected cells by searching for these help me or SOS signals. And when they find an infected cell, they release their killing molecules. these molecules will actually destroy the infected cell and everything inside the cell, which essentially shuts down the virus factory. Antibodies also reach the lung from the blood and attach to the sticky spike protein, which blocks the ability of the virus to get inside the lung cell, stopping the infection. So, while these strategies can be very effective at removing virus from the body, they have one critical downside. It takes time for these cells to get ready to fight. And while these cells are training, the virus is continuing to spread throughout the lungs. By the time the specialized immune cells are ready to fight, the lung is full of virus. The T cell sees all of these infected cells and starts killing them, which can actually lead to significant damage in the lungs. Ideally, the immune system would be ready to fight before the infection has spread this much to avoid such extensive damage. And so to fight an infection like SARS-CoV-2 more effectively, we need to give the immune system more time to train. How can we give the immune system the head start that it needs? So remember, during an infection, the first responder cells are educating the specialized immune cells about the invading virus while the viral infection is ravaging the lungs. Vaccines are a way to provide this education, provide this information about the invader in advance. And so vaccines contain what is called an antigen. An antigen is just a piece of the virus, not the whole virus, just a piece that can stimulate or activate the immune system. And in this example vaccine here, and most vaccines for SARS-CoV-2, the antigen is actually this sticky spike protein. Exposure to this protein actually allows the immune system to learn how to fight this virus in case of a future infection. Choosing the ideal antigen or the best piece of the virus to educate the immune system can take years of research in order to design the most effective vaccine candidate. And so we can imagine what would happen in a vaccinated individual. So they could be exposed to SARS-CoV-2, and that virus can enter the lungs and maybe infect a cell or two. But the immune system doesn't have to waste any time training. The T cells can enter the lungs right away and eliminate these infected cells rapidly, shutting down those virus factories. And antibodies can block up all the viruses outside of cells, keeping the lungs healthy. And so in the next section, we'll talk a little bit more about the modern vaccine designs being used right now to fight SARS-CoV-2. But again, I want to summarize and pause for some questions. So I told you in this section that SARS-CoV-2 infects and damages lung cells, converting them to virus factories. Lung cells can call to the immune system for help. The first responder cells hear this call and recruit specialized immune cells to join the fight. T cells, which are a type of specialized immune cell, can destroy infected lung cells and shut down those virus factories. Antibodies, which are produced by B cells, can block the virus from entering and infecting more cells in the lungs. And lastly, vaccines work so well because they give the immune system a head start, which means that the immune system is ready to fight as soon as it's exposed to the virus without any delay. So I'm happy to answer any questions on this section if we have any.
Thanks, Allie. We'll start with a couple questions from the YouTube chat. They first point to the updated CDC guidelines for a 10-day quarantine that pinpoints the highest rate of transmission between days 3 and 4 and day 14. I'm not sure exactly the source of that data, but I did know that the CDC had updated those guidelines.
Yeah, so this happened today, actually.
Yeah, it happened earlier today.
Yeah.
Science really in the news. So the questioner is asking, wouldn't that mean the risk of transmissibility is low at first infection before it has replicated enough in your body to be able to be detected by the test? Same as when you test negative again.
Yeah, so it's true that having such a low viral burden that you're testing negative means that you're unlikely to be shedding a large amount of virus into the population, but it's still possible. And we can see that because asymptomatic people can infect others. But it's the people who are infecting the most others are those who are likely spreading the disease when they aren't sure that they're sick yet. So I think there's sort of two thoughts here is how long do you have to quarantine? 10 to 14 days, whatever the new recommendation is. And then when are you likely to test positive for the first time? And that happens around day 5 or 6.
Fantastic, thanks. And one other question. So if viruses don't survive outside the human host for long, why do we consistently struggle with seasonal colds and flus? Are they just simply always active in some other area of the world?
So that's a really good question. So you're right. When the US is experiencing summer, other parts of the world are experiencing winter and their flu season. So that's how we predict what the flu will look like each year because it changes and develop a vaccine. So we look at countries that are experiencing winter while we're experiencing summer, and we can guess what flu vaccine or flu strain is going to be the most dominant for this year. But for coronavirus, there's not really much evidence that the virus is changing in the same way that the flu does. So I don't think we will have to approach it in the same way.
All right, we have a bunch of questions. I'll move to some Zoom questions now. So John is asking, what is it about aging that makes older people more susceptible or vulnerable to coronavirus?
Yeah, this is a really interesting and really hard to answer question. There is a lot of active research being done on this right now. And the short answer is that we just simply don't know. There's something about aging that makes the immune system less responsive, meaning it either doesn't get educated as quickly or it can't use its tools as efficiently to remove pathogens from the body. We know that it happens, we just don't know why. And so I think that will be a really important area of research moving forward, is trying to understand what exactly is the block or loss that happens with age, so we can attempt to boost the response or at least, supplement it in some way to make folks less susceptible to infections.
Thank you for that. Another question is, how do B and T cells know how much to produce?
Yeah. So so what you're kind of hinting at here is there's actually so many layers of regulation and interaction that I didn't even describe, but there's a lot of cells in the body, immune cells, whose job it is to regulate the activity of the immune cells that have these big guns. Because as you can imagine, we don't want an active immune system all the time. We want the immune system to be activated, to clear the infection, and then to return back to normal. And so there are other cells like regulatory immune cells that part of their job is to kind of sense what's happening in the body and shut down the cells that have these powerful machines. But an important part of this is that those two cells, those B and T cells that have these big guns, they also create forms of themselves called memory cells. And these cells stay in the body. They're not producing any, they're not actively doing anything. They're just waiting until an infection happens again, say later in life. And then they can become reactivated and respond to the infection more quickly. So hopefully that answers your question.
Thanks. And again, if anyone has follow-up questions, they can put that in the chat too. We have another question from a YouTube viewer who's curious for someone like themselves taking immunosuppressants, asking how effective might a vaccine be for them, or if there's any interplay there.
Yeah, that's a really good question. And my answer is that I'm not a physician, so I can't offer medical advice. And that's something that needs to be discussed with your doctor. But for folks who are immunocompromised, there always is that interplay between maintaining the status of controlling whatever syndrome or concern there already exists, and then enabling the body to fight the infection with a vaccine. And that is very complicated and depends a lot on the type of pre-existing condition. So talk to your doctor about that. Yeah.
Great. And I think a somewhat related question asking, do some people get hyperactive immune responses that then causes different problems?
In response to like vaccines or to SARS-CoV-2
In
general?
The questioner doesn't specify, but I would assume it's in response to COVID-2.
So the immune system is very good at destroying cells that are infected. And so you can imagine that in a lung which is full of virus, these T cells are just coming in and destroying a ton of lung cells in order to try its best to shut down these virus-making factories. And so a lot of the damage that we see during these infections is actually caused by the immune response trying its best to win a losing battle. And so depending on your definition of hyperactive, that could be, I guess, in that category. But you know, your body's really good at doing its job, and that's why vaccines are so effective, because it enables them to figure out how to do it a little bit faster. So when the virus comes, it can just go in and eliminate it right away. There's no lag time.
Yeah. And then one more question from a Zoom viewer: how long does that immune response last?
Yeah, so because it's a new virus, we do not have like long-term data on how many people are, you know, continuing to stay immune. I think the longest information we have right now is that people maintain immunity for up to 3 or 4 months. And we're just going to have to continue to monitor people throughout time moving forward to see how long that immunity lasts. It's just too hard to predict with such a new virus. But I think that the people they've enrolled in these trials, you know, 5 months ago, so the data is like 3 to 4 months the immunity lasts.
Thanks very much. Are there any other questions?
Do another one, and then we have like a little 5-minute break after this, right?
Yeah. Yes.
Okay, I'll read Olivia's question. Is the time needed for immune cells to get ready to fight predictable? As in, does it take the same amount of time for everyone, or is it different for each person?
Yeah, so there's an average of probably 2 weeks to mount a effective immune response with the specialized immune cells, but certainly some individuals will respond faster, and maybe older individuals or those who are immunocompromised would respond slower, if at all. So absolutely, there's variety, but on average, it takes at least a week, up to 2 weeks, for the immune response to be fully functional. And that's probably why we see different severity of illness too, because whenever that time window is between the virus getting out of control and the immune system being fully activated, there could be a time window there of controlling the disease. Yeah, so let's pause here for a short break so we can stretch and have some water, and then get back to it. And we'll talk about the new vaccines that are being produced by companies around the world and how they work.
Hello, everyone. We will be getting started. Hope you're there. And this is our third and final section. And then we'll have questions at the end, as usual. And then we will be sending you a quick feedback survey, and we would really appreciate it if you filled it out. I'll remind you at the end too, but so that you don't disappear in the end. But yeah, take it away, Ali.
Cool. Welcome back, everyone. It's good to take a little break from Zoom sometimes. So next, we're going to be talking about the current state of vaccine research for SARS-CoV-2. So before we get into any specifics about how these vaccines work, I just want to spend a few minutes thinking about the properties of an ideal vaccine candidate. First and foremost, a vaccine must be both safe and effective. This means that they should induce minimal negative side effects and provide protection against actually getting the disease. Additionally, we would prefer if a vaccine was easy to store and distribute so it can be rapidly delivered around the country and around the world. And currently there are approximately 200 vaccines in various stages of development. And the next few slides, I'll walk you through the stages of developing a vaccine and how many candidates are at each step. So the first stage is preclinical development. There's over 150 vaccines at this stage. And this type of research is usually conducted over many years to try to figure out what piece of the virus best stimulates the immune system, to try to figure out what the best antigen is. What parts of the virus can we actually safely use? And at this stage, the scientists are usually conducting experiments with cells in a dish, looking at the virus under a microscope, or even testing vaccines in animals to try to figure out that ideal antigen. And once the antigen is chosen, vaccines can be produced for clinical trial testing. Currently, there are 15 vaccines in phase 1 clinical trials. These trials involve small groups of healthy volunteers. These volunteers receive the experimental vaccine, and doctors and scientists monitor them very closely for any side effects, and also test their blood to see if their immune system is developing antibodies, which is a sign that the immune system has become activated and is ready to fight the virus. If the vaccine seems safe and stimulates an immune response, it can progress into phase 2. Currently, there are 14 vaccines at this stage. Phase 2 clinical trials are much larger than phase 1 with many more diverse volunteers. These volunteers all receive the vaccine and again are monitored very closely for safety and if they develop antibodies. The last type of clinical trial is phase 3, which tests if the vaccine protects against disease. Currently, we have 11 vaccines in phase 3 trials. These trials typically recruit thousands of volunteers who are split into 2 groups. One group receives the vaccine and the other group does not. These volunteers then go live their life, and then scientists see at the end of a determined time period which group has more infections. If the vaccine is working well, the individuals in the vaccinated group will be protected from infection, and that group will have less disease than in the non-vaccinated group. So there are several companies who in the last week or two have announced very positive results from their phase 3 clinical trials. And here I'll list the 3 that have gotten the most media attention. So first is a vaccine from the company Pfizer in collaboration with the company BioNTech. Their phase 3 clinical trial was tested in 45,000 volunteers, and they found that it was 95% effective at preventing the disease. Volunteers reported tolerable side effects, which included headaches and some soreness at the injection site, which is really promising news, so nothing too negative. And the one downside of this formulation, unfortunately, though, is that it requires storage at ultra cold temperatures, so minus 95 degrees Fahrenheit. And as comparison, a normal freezer in your home is approximately 0 degrees Fahrenheit. So you can imagine it would be really costly and difficult to transport this vaccine, to store it, and to distribute it around the country. Another promising candidate comes from the company Moderna in collaboration with the National Institutes of Health. They tested their vaccine in 30,000 volunteers and found that it was over 95% effective at preventing severe disease. Similarly to the vaccine created by Pfizer, the Moderna vaccine volunteers reported very minimal side effects. But excitingly, this vaccine is much easier to store. Ideally, for long-term storage, it would be stored at minus 20 Fahrenheit, which is actually a much more reasonable temperature for lab freezers to reach. But for short term, it can be stored at 40 degrees Fahrenheit, which is a temperature of a normal fridge. And lastly, the company AstraZeneca, in collaboration with the University of Oxford, has recently released results that their vaccine is 70% effective, but we still don't have as much information about this trial yet. And so after such promising results, companies like Pfizer and Moderna would next apply for approval for their vaccine design from the FDA. And part of this review process includes a rigorous analysis of all the data the company collected during their clinical trials. And this data is reviewed by an independent board of scientists and physicians, which are not associated with the company, to determine if the vaccine is acceptable for use. After approval, the vaccine is also continuously monitored to ensure that it remains safe. So The time from preclinical studies to distributing a vaccine to the public is typically pretty slow, averaging about 10 years. And I'll explain to you the reasons why this takes so long. So first, studying the virus in the lab to find the ideal antigen for the vaccine can take many, many years. After the candidate is selected, clinical trials are run consecutively or one after each other to test the vaccine for safety and efficacy. And then if it works, it gets approved. And at this point, the government will invest in the very expensive and time-consuming job of building these factories to manufacture millions of doses of a vaccine, which can take years. So there's really two major slowdown points to get the vaccine to the public. The first is to find the ideal antigen to educate the immune system. And the second is actually building the infrastructure to make the vaccines once we have an approved design. So you may be wondering, how did we get to this point where vaccines are predicted to be available for COVID as soon as next year? And I want to convince you in the next few slides that this process actually happened in a very safe way. So first, and arguably the most important way that scientists sped up this process is that we already knew the ideal antigen for a vaccine at the start of the pandemic. Scientists have been studying coronaviruses SARS and MERS for 15 years. And one incredible example of how this early research paid off is the work of Dr. Corbett, a viral immunologist at the National Institutes of Health. Before the pandemic, she led a research team at NIH that studied coronaviruses, SARS, and MERS. The focus of her research for many years was to find the coronavirus vaccine antigen. She realized that of all the parts of the virus, this sticky spike protein was the ideal antigen to educate the immune system on how to fight the virus. When SARS-CoV-2 emerged, she brought this information to Moderna and worked with them to develop their vaccine. Because of her work and her investment into researching coronaviruses, when the pandemic hit, we didn't have to spend 10 to 15 years trying to figure out the ideal antigen, which is pretty incredible. So we were already starting ahead, but another time-saving strategy scientists used was to run clinical trials simultaneously or somewhat staggered to save a little bit of time. But the other most important time saver was that we invested in building the factories and creating the infrastructure to make these vaccines before one is approved. This way, the factories are standing ready, so as soon as a vaccine is approved, it can immediately be manufactured and distributed. And so this means that scientists conducted the preclinical research and clinical trials with the same rigor, but investing in the infrastructure now instead of waiting will end up being a huge time saver. So another feature of the Pfizer and Moderna vaccines that's so cool is that they're using a new strategy to deliver the antigen, which saves some time in the lab. So traditionally, vaccines are formulated to include this piece of the virus or antigen, which cannot cause disease, but is used to educate the immune system. And if you recall from earlier, that process goes something like this. The first responder cell will find this antigen in the body and present it to the specialized immune cells, who then get ready to fight the virus by creating their killing molecules and antibodies. But what is very hard and time-consuming about making a vaccine like this is that you have to make the antigen in the lab, which can be really difficult. Scientists have to figure out how to make these proteins from scratch, figure out how to keep them stable, and how much of it needs to actually go into the vaccine. And so for many years, scientists have wondered, is there a better way to actually deliver these immune-stimulating antigens into the body? And very recently, the technology has advanced to enable scientists to try a new approach, giving the cells instructions to make the antigen themselves. And so let's remember what happens during an infection so we can see how this new technology mimics infection in a safe way. So in an infection, SARS-CoV-2 enters the lung, attaching it to the lung cell with that spike protein, and delivers genetic material containing instructions to make the virus into the lung cells. The lung cells then become virus factories, reading these instructions and making more viral components, which then assemble into new viruses that can then repeat this cycle in the next cell. And remember, these infected cells know that these are foreign materials and can call to the immune system for help. In this new technology, instead of delivering that antigen directly, we're rather giving the cell just a snippet of that genetic material it would see during the infection. This small piece of genetic material called mRNA, the cell can read and use it to make one piece of the virus, the antigen that we are interested in. This antigen is then presented on the surface of the cell and can be used by the immune system to train for future infection. The reason this is really exciting is that mRNA vaccines have a lot of features that we want. They're really safe. The virus can't form. We're just making this antigen. The mRNA instructions are actually very short-lived. The cell breaks them down and gets rid of the mRNA quickly after making the instructions, so there's no long-lived antigen around in the body. And importantly, this process actually is mimicking what happens in a natural infection. The antigen is produced inside cells like it is during infection, and this can provide a really strong and robust education for the immune system. So this new technology allows us to develop new vaccines really quickly. Once the ideal antigen is known, it's very easy to produce these snippets of genetic material, these instructions in the lab. And this technology could be used in theory to deliver instructions for nearly any antigen you can imagine without having to do a lot of time-consuming optimization. And so in our last few minutes here, I want to leave you with a sense of the time scientists have invested in creating this vaccine over the last 15 years. So in the mid-2000s, after the first outbreak of SARS, scientists began studying coronavirus in earnest to find a vaccine antigen. The information came in handy when SARS-CoV-2 entered the scene. Scientists identified that it was a coronavirus and then quickly published the full set of viral instructions. Because we already knew the antigen target and we have this really fast mRNA technology that didn't require optimization, it only took 2 days for the first mRNA vaccine design to be completed. And by mid-March, volunteers were being enrolled in clinical trials and receiving the experimental vaccine. And just a few weeks ago, we learned that these vaccines are very effective. So looking ahead, Pfizer and Moderna have applied for emergency authorization from the FDA, which facilitates a rapid review of the clinical trial data. And likely healthcare workers and other vulnerable populations will be the first to receive the vaccine, with the rest of the country sometime after, hopefully next year. And while we wait, we can continue to protect ourselves by wearing masks and continuing to social distance. So in summary, investing in early research can have a huge payoff, and sometimes in unexpected ways. So 15 years of coronavirus research before this pandemic really helped us design these vaccines and be able to implement them super quickly. We already knew what the target antigen was at the very beginning of the outbreak. I told you how we're building the factories and facilities to actually produce these vaccines earlier instead of waiting for approval to start that process, which again is another huge time saver. And the most exciting part is that we have a new design of vaccines, mRNA vaccines, which allow for delivery of instructions to make that antigen, make that piece of the virus, which effectively educates the immune system. And of course, I have to acknowledge Science in the News for their support, and I am happy to take any questions on this last section.
Thank you so much, Ali. We have so many questions. But in case some people are leaving, I'm going to post a link to our survey. It's very short, it's 1 minute, so that we can get feedback for our lectures that will start in the beginning of 2021. This was the last lecture of the year. Should we start from the ones that are on YouTube, Lily?
Sure, yeah. One question we got earlier was basically following up on if older people are responding more slowly to COVID. Is there any sense in which the vaccine could be more dangerous to them, or a vaccine?
The vaccine does not contain any virus. It is impossible to acquire the infection from the vaccine. So I would say as long as your physician considers you to be healthy, then there shouldn't be any risk from the vaccine to get the infection. So does that answer the question? I'm not sure I totally understand.
Yeah, I think that that answers the question.
Yeah, I think the most important thing here is that, let me show this picture again. This cell is producing one piece of the virus, and this is not enough to cause an infection. It's only enough to educate the immune system about what's happening. But specific interaction between like individuals who are immunocompromised in the vaccine, I'm not qualified to answer that.
Great, thanks. And one other question we had was asking, aren't there other tools for immunity in the adaptive immune system other than just antibodies?
The main two tools are antibodies and these T cells, which kill the infected cells. Those are the two major players. There are other cells that respond earlier and can help stop the spread of infection while the body's waiting for these more powerful cells to get activated. But these are really the two primary jobs.
Great. And then a question on the timeline that you had for the stages of the vaccine is how long do the usual phase 1, 2, and 3 clinical trials last?
So phase 1 and 2, like 2 years maybe, and then phase 3, I'd say like 3 to 5 years. And a lot of that is because it's sort of iterative. So you release data to the FDA or these organizations that approve the drug, and then they'll ask you to go back and do more trials, and it continues on like this. But for an emergency situation like this, it's possible to speed it up in a safe way and just continue monitoring as we move forward. Thankfully, the vaccines people have reported have like really seem to be protecting people extremely well, like 95% effective. So they seem to be working great.
Perfect. We have some, now we're moving to some of the Zoom questions. One is, who developed the mRNA technique, and is it available to all, or is it licensed?
Say that again.
Who developed the mRNA technique, and is it available to all?
Yeah, so the technology itself is patented, but different companies have patents on like different formulations or different versions of how exactly they're delivering this genetic material into the cells, which is why you see differences in like what storage temperature they require and differences in efficacy. And so it's a little bit complicated because it's hard to think about patents with biology because you can't patent something that exists in nature, but you can patent something new that you created. So I believe that each of these companies has their own patent on their specific formulation. But in theory, anyone can make an mRNA vaccine, or any company, I should say, but they would have to have a unique formulation.
Perfect. Tracy was the one who asked the questions, and he says thanks. Another question is from John. Are there possible interactions between the different vaccine candidates? Let's say 70% of the 70% one is the first to get released. Should people be judicious about which vaccine they decide to take if they have a choice? Lest a better one come along later and they can't have it because it will cause an interaction problem with the first one, too much of an antigen or whatever. So does it affect it?
Yeah, that's a really good question. And I am honestly not sure of the answer to that. And I think, truthfully, it depends a little bit on what the antigen being used in the vaccine is. a lot of these vaccines are going after using the spike protein because it's the one that was available, we knew would work. And I imagine that's an interesting question. I'm not sure the answer to that. I would guess that there wouldn't be, because it's just would be like a boost and you would just, like, your body already is responding, so it might not change anything. But yeah, I honestly don't expect this. I doubt that this 70% effective one will be the one that ends up getting widely distributed, because there's others that are more effective. Yeah, that's a tough question, but.
Yes. Okay. Another one is, what about the other 8 vaccines in the clinical trials? What technology are they based on? Because we only talked about the mRNA ones.
Yeah, so traditional vaccine design can take advantage of just injecting the antigen directly, or people can actually take the virus and kill it or weaken it and use that in the vaccine formulation as well. Those are the primary 2. And the other 8 or so vaccines are formulations with that type of technology. In the handout, there's a link to the New York Times and Washington Post vaccine tracker, where they go through like all of the major companies that are making vaccines and what the different formulations are. So if you're interested, you can read more about it there.
Perfect. Thank you. Another question is, we know you're not like a logistics expert of vaccines, but who's building the factory? Is it Moderna, Pfizer, someone else? Is it government related? Is it in countries? Where?
Yeah, that's a good question. I think of course the government has some role in that, but they're being built in partnership with the company that is, making the vaccine. So I think I read a press release about Moderna partnering with the government, or getting some like $500 million grant like months ago to start building these factories. So I'm sure that the people who invented it have to be involved in the production of these facilities in order to them to meet the standard that they would require to actually assemble the vaccine to their specifications. So I think it's a multi-group effort.
Let's see, we have a lot of questions. Another one is the people who are in the trials. How do we know they were exposed to COVID? Were they exposed to COVID? Because if they didn't get infected, maybe they were just not exposed.
Yeah, so this is a question about statistics. So thinking about this experiment, there's 30,000 people in this trial. 15,000 of them got the vaccine, 15,000 of them did not get the vaccine. And then you wait. And it is improbable that all 15,000 of the vaccinated people would not get the infection simply because they weren't exposed. You have to assume that the 2 groups are being exposed to the virus at a comparable rate or comparable frequency. And that the reason you're seeing more infections in the non-vaccinated group is because those people are not vaccinated. So in clinical trials where there's such large groups of people, you can assume that the 2 groups are essentially equivalent in terms of exposure to the virus. And the only difference between them is the intervention you give, like in this case, a vaccine.
Yeah, thanks. And I think there's a related question that just popped up on YouTube, which is, how did the increase in number of cases of COVID-19 impact the speed with which they were able to do the trial?
Yeah, so they can calculate how many cases they need to see in order to have like statistical rigor in comparing the 2 groups. And if you have more active spread in the population, you just reach that number you need faster. So I think they needed to see like 200 cases in their group of 30,000 in order to effectively evaluate if the vaccine was being effective or not. And that's sort of the preliminary data that has come out. Of course, they'll continue to monitor this over the course of the coming weeks and months to see what the longer term effects and protection is. But yes, I'm sure that the increasing spread of the virus contributed to them being able to find the answer more quickly.
Perfect. Our next question is again about these new mRNA vaccine technologies. How long has it been in development, the mRNA technology?
So Moderna has been a company for 10 years. They were founded in 2010, but scientists have considered this strategy for years, maybe even decades before that. The technology to deliver the genetic material just wasn't there. Let me scroll to this slide. So RNA is very fragile, and you can't just inject it into cells. It will just get destroyed right away. So they needed to encase the RNA in some type of fatty coating in order for it to be delivered to the cell and not be destroyed immediately. So developing that technology is sort of what Moderna was founded on. So even though this idea has been around for a long time, we didn't really have the technology to put it into action until the last 10 or so years. So a lot of things kind of were happening simultaneously with this pandemic to help us, before this pandemic, I should say, that helped us make something effective really quickly.
Perfect. Another question about vaccine development in general. The advances that vaccine development has been undergoing this entire during this pandemic, do we think that it's going to help the development of future vaccines for future?
Yeah, absolutely. So this process is really fast. Like I said, if you're actually giving people the antigen protein, you have to spend a lot of time figuring out, how do I keep this stable? How much protein do I give? If you want to give them the genetic code to make that protein, it's literally copy paste. So in theory, you could use this vaccine strategy to rapidly make a vaccine for almost any antigen. And there's even ways to use it to stimulate the immune system for cancer and other types of diseases. And companies like Moderna and Pfizer are pursuing those now.
Perfect. So another question about, let me see if there's a more relevant one. Well, we can go back to the questions about the elder population. Older people take a stronger flu vaccine for the general flu. Is there likely to be a special formulation for the COVID vaccine for the over 65 group?
That's a good question. So the expanded clinical trials, let me scroll to the slide, sorry. So the phase 3 clinical trials that test, 30 or 40,000 people, they usually try to sort of recruit people of diverse ages, races, different genders, et cetera, to make sure that they can assess how different types of people are responding to the vaccine. And I haven't looked at the data closely enough to know what the robustness of the response is in people over 65, but I would assume it's not as effective as those who are younger. I haven't heard information about a specific formulation being made for older populations, but it's something that I think they could consider. But honestly, we're still collecting this data as we speak. So I think it's just going to be moving forward, waiting to get robust data from all populations. Yeah.
Well, speaking of populations, so, like you said, there was a 95% success, right? Do we know anything specific about the 5% that did in the end get infected that were vaccinated? Is there commonalities in terms of age, immune system function, or other illnesses?
Yeah. So that data I don't think has been released to the public yet. But what is very promising about the Moderna vaccine is that they claim that even in people that got COVID in the vaccinated group, that it's 100% effective at preventing severe disease, which means that the people who were infected after receiving the vaccine, their symptoms and the disease they experienced was much less severe than it would have predicted to have been otherwise. So we don't know a lot about that information right now, but as we move forward in time, more of this data will become available to the public, and scientists can, of course, assess it and ensure that it's safe.
And another question where the data might still be in progress, but the question is, how would the effect of the vaccine come into play if the person already has the virus?
If a person has the virus, I don't think the vaccine would help them necessarily, because your body still takes that 2 weeks to mount a response when you're vaccinated. You're just mounting that response ideally when you're not infected. So if someone's already has the active infection, their body's mounting a response, and they get vaccinated, their body's mounting a response. So I don't think it would speed it up for people who are actively having the infection. So ideally you get it before you're exposed.
All right, this looks like our last question, but if you have more, please post them. It's from Anthony from Zoom. How reliably can mRNA be designed or controlled to produce only the intended antigen? Are there risks with this type of treatment that in the presence of certain body chemistries, the mRNA might erroneously trigger the production of unintended harmful products in cells?
It's a great question. mRNA is incredibly specific. It makes one protein. And this mRNA is designed to make this one viral protein, and it can't make anything else. So no, there is not a risk of off-target production of other virus proteins. And in fact, the mRNA is cleared from the body really quickly. So it comes in, the cell makes the one protein that it's designed to make, and then the RNA is eliminated.
Perfect. Another question about the vaccine and being infected. If you have had, if you were infected by COVID in the past, but you didn't know, you were asymptomatic, you weren't diagnosed, does that cause any problem, or does it just mean no benefit from the vaccine?
So if someone was infected in the past, you could tell by checking their blood for antibodies, which should last in their system for, we know, up to 3 months, but hopefully longer. And I think that there were people who had this situation in some of the clinical trials, like for Moderna. And I don't believe there was these individuals who had previously had the infection and didn't know, they volunteered, enrolled in the clinical trial, received the vaccine, and I don't think there were any issues observed. So I think it should be fine. But again, with all of these answers, the caveat is all the data has not been fully released yet. So we'll have to review and keep assessing and making decisions based in evidence as the information becomes available.
Perfect. That looks like all of the questions. I will post in the chat our link to our very short 1-minute survey again to get a sense of what kind of talks you're interested in for the future, what you thought about this one, how we can make it better. But thank you so much for joining us. Allie, thank you so much. This is so helpful, so timely. We haven't had a talk that's this relevant to our daily life in a while. So
hopefully it was helpful, and everyone, please stay safe. And again, I want to reiterate, if you're experiencing symptoms, so please contact local health authorities for guidance and check out the mass.gov and cdc.gov coronavirus websites for more information. And in the handout for this talk, there's also a bunch of links on different types of tests that exist and just kind of considerations for quarantining if you need more information. So thanks, everyone.
I posted the link to the handout again. That has all the links Allie forwarded us to for further info if you want to keep learning more. Thank you, everyone.